Hello everyone. In the second chapter that we have in SAT, it's about the cell. You know that the cell is the basic unit of life. Okay, as you know that our body made up of many organs. When we have a disease or when we have some difficulties or abnormalities in any organ of our body, in order to understand them, how they are working, what are the reasons behind those diseases, how is their physiology, we have to understand their basic structures, which all of them they made up of cells. So the basic definition for the cell is the basic unit of life. Okay, and you know that the cells are made up of different organelles, and each organelle has its own function. Generally, we have two types of cells. We have eukaryotes and prokaryotes. Eukaryotes, they have more complex structures, and they have membrane-bound organelles. They are found in animals, plants, fungi, in those kind of organisms. While prokaryotes, they are like primitive structures. They are not such developed like eukaryotes. They have simple structures, okay? And they don't have any membrane-bound organelle. An example for the prokaryote is bacteria. So, guys, now let us start by solving those questions. As you see, we have those organelles. We have smooth endoplasmic reticulum, okay, in which its function is breaking down of alcohol and toxic materials. Nucleolus, it's found inside the nucleus, and its function is production of RNA. Ribosomes are found inside the cell, and their function production of or synthesizing of protein. Lysosomes are organelles that they are found inside the cell. Mostly they are found in animal cell, and they contain enzymes, and their function is fighting with bacteria and viruses. They have protective function, that they protect our body. And we have Golgi apparatus, is an organelle that's like it's attached to the smooth endoplasmic reticulum, or maybe it's not attached. Its function is processing, modification, and packaging of proteins. So, in the first question, it says the site of protein production. We had the answer for this one is ribosome. Okay, so it means C. Found in large quantities in white blood cells. Guys, white blood cells function is protecting our body from microorganisms, from parasites, from bacteria, from viruses. So they should contain enzymes that they are fighting and digesting those microorganisms. So white blood cells are full of lysosome, or they contain lysosome and other phagocytic cells. So the answer for this question, in which white blood cells and phagocytic cells, they contain large quantities of lysosome. Okay, so the answer for this question is lysosome. D. The site of RNA production, guys. RNA is produced from the nucleolus. So the answer for this question is B, which it is nucleolus. Okay, I think they are clear. In this question, It asks for the diameter of eukaryotic cell, or it asks for, we can say, for the size of a eukaryotic cell. Guys, generally, the eukaryotic cell sizes are about 100 micrometer, or it may be 200 micrometer. They are in those ranges. So the answer for this question is B. Okay, about 1 nanometer and 10 nanometer, these are generally the diameters for viruses. Okay, viruses are so tiny, they are very small. We cannot see them by the light microscope. Okay, we have to use electron microscopes in order to see them. They are in this range. So nanometers are used for virus, as I told you. 1 micrometer, or from 1.2 to 2 micrometer, or from 0.5 to 2 micrometer. In this range, the micrometers are for their sizes or diameters for prokaryotes. Okay, prokaryotes. Or we can say these are the diameters for bacteria. So the answer for the question, diameter of a eukaryotic cell, is B, 100 micrometer. So we have all of the following are true of the electron microscope, except, so you know that electron microscopes are magnifying the images for us about 100,000x. Okay, they are magnifying the images for us that we cannot see by the light microscope. So let us read them. The resolution is greater than that using a light microscope. Yes, the resolution, its magnification power, is greater, so this is true. The magnification can be greater than 100,000x. Yes, it's true, guys. Its magnification power is about this range. Living specimens can be studied in great detail. No, this is not true. For electron microscope, we need many chemicals. We need many equipment. We have to take a small segment of the cell, a small specimen. Okay, so we have to cut them, and the cell will die. Actually, guys, we cannot use living specimens or living samples for the electron microscope. So this is the answer, but we have to read the others. A narrow beam of electron forms an image. Yes, this is true. For electron microscope, we are depending on the beam of electrons, while in the light microscope, we are depending on the light source. Okay, so the beam of electrons, the source of energy or light that we are using. Okay, it takes hours. Yes, guys, the preparation of these specimens or slides for the electron microscope, it requires hours to prepare a specimen for viewing. It's not like a light microscope that we can prepare a slide in 5 minutes or 10 minutes. Okay, but for electron we need nearly hours, maybe days. We need to prepare our slide. So the answer for this question is living specimens that can be studied in great detail. This is not true. Dead cells or dead specimens will be used. So the answer for the question is C. Okay, let us move to the next question. All of the followings are correct about prokaryotes, except, so all they are true except one of them. They have one chromosome. Yes, guys, we know that. Prokaryote, they have only one chromosome. They contain ribosome. Yes, exactly. They contain ribosome, but the size of the ribosome is different from the eukaryotic one. They are smaller than eukaryotic cell. Yes, we mentioned that. They are smaller than eukaryotic cell. They can be either anaerobic or aerobic. Yes, we have some bacteria or some prokaryotes, they are aerobic. It means they require oxygen. Okay, anaerobic, they can live without oxygen. So we have some microbes or some bacteria that they are living in the soil without the presence of oxygen. They can live. Okay, and then we have some that they require oxygen. So they are true. They contain small mitochondria. We know that prokaryotes do not contain mitochondria. Okay, so the answer for the question is E. They contain small mitochondria. This is not true. It's wrong. They do not contain mitochondria. You know that the function of mitochondria in eukaryotes is responsible for the cellular respiration. And it's a double membrane organ. It's a membrane-bound organelle. We don't have such membrane-bound organelles in prokaryotes. Okay, so but just know that for your information, prokaryotes, they contain the enzymes freely inside the cytoplasm. The enzyme that they found inside the mitochondria. Okay, they contain the enzyme that they are found in the mitochondria inside their cytoplasm, but they do not have mitochondria. So this is the answer for the question. Okay, let us move to the next question. The phase contrast microscope. Okay, let us see which of the answers is related to the phase contrast microscope. Uses a beam of electrons to form an image. This is not true. Electron microscope uses a beam of electrons to form an image. Developed by Robert Hooke, and it says developed by Anton van Leeuwenhoek. These are not true, guys. During the time of Robert Hooke and Anton van Leeuwenhoek, they developed compound microscope, the first form of the microscope. Okay, it's commonly used to study genes. For studying genes, we cannot use microscopes. Okay, in order to study genes or the sequence of the genes, we have used DNA sequencing. Okay, by sequencing method, we can identify the nucleotides. Or by studying PCR, we have polymerase chain reaction. By this equipment, we can use the studying of genes. Okay, PCR, gel electrophoresis, these are the techniques that they are used in the molecular biology to study the genes, specific genes that we want. Okay, it's useful in examining unstained living tissue. Yes, phase contrast microscope, it's used for studying or examining the unstained living tissues. Okay, for unstained living tissues without using any chemical, any substance, any staining material, we can study them by the phase contrast microscope. So all the others from A to D are incorrect about the phase contrast microscope. Question 9. Which would be best to study the structure of thousands of mitochondria? So guys, we know that mitochondria are organelles that are found inside the cell. So they are smaller than the cell. Okay, and we need to study the structure. It means the structures and the details of the mitochondria, which it's found inside a cell. So how we can study it? You know that we cannot study it by the light microscope. we have to study it by using transmission electron microscope. Okay, transmission, it means the beam of electrons passes or moves into the cell, into the mitochondria. We can study it, we can see it. Also, we have scanning electron microscope. Know this for your information. Scanning, it means it's just used for studying the surface. So we cannot use a scanning electron microscope. Also, light microscope, we cannot use it. Light microscope, it doesn't have such ability to magnify the cell. So how we can study it for the mitochondria, which is very tiny organelle found inside the cell. So the only way that we can study them is by using transmission electron microscope. Okay, so the only way, as I told you, in order to study is transmission electron microscope. Let's just read the possible answer that we have. Dissecting instruments and scanning electrons. Scanning cannot be used, guys. We have to eliminate them. Okay, we have to skip the answer that they are not they don't have any relation. So ultracentrifuge and scanning, they are not used. Dissecting tools and light microscope. Light microscope doesn't have such ability to magnify the mitochondria. So the only thing that we can use it is transmission electron microscope. Transmission. Okay, let me change the color in order to be identified. Transmission electron microscope and transmission electron microscope, but by dissecting instrument or ultracentrifuge. Guys, dissection, which you know that it's like cutting and taking a segment or a specimen. It's not such way to be used by the transmission electron microscope to see the mitochondria. We have to use ultracentrifuge. So the answer for the question, guys, is C. Ultracentrifuge, it's centrifuge that's used in the lab for separating the organelles that we have, different organelles in the cell, and each organelle has its own density. So the ultracentrifuge, it separates or makes layers, okay, according to their density, that we can isolate the mitochondria, and we know that mitochondria has its own density. We can find it, and after that we can use transmission electron microscope. We can study the structure of mitochondria. Let us go to the next question, which is the last question in this part. In the part 2, in the next part, we will continue from the question 11. So, what would occur if a plant cell were placed in a strong hypertonic solution? So, guys, we have a strong hypertonic solution. When we say hypertonic, it means high solute. When there is high solute in a solution, it means low water. The amount of water is low. Okay, so this is the solution. What about the cell? When the outside is hypertonic, which it means when it is high solute and low water. This is outside. What about the cell, which it is a plant cell? Okay, so this is the plant cell. Let me write, this is the plant cell. So when the outside is hypertonic, inside the cell it will be hypotonic. Hypotonic, it means low solute. I will write by LS. It means low solute. So when solute is low, the concentration of the solute, it means high water. Okay, HW, it means high amount of water. And if you come to the definition of osmosis, which it says, osmosis, guys, we know that osmosis, let me write the definition of it so you can understand. Osmosis is the movement of water from high concentration, I will write it by HC, it means high concentration, to low concentration. Okay, so where water is high, guys, as you see, inside the cell or outside of the cell. As you see, inside the cell there is high water. Okay, and we said that the movement of H2O from high concentration to low. So water moves from high to low, in which the outside is low. So when the water comes out from a cell, what will happen, guys? When the water comes out, you know that the cell will be shrinked like this. It will be smaller in size. Okay, because water came out. So let's read the possible answers. It says nothing would happen because nothing would happen because the cell wall would prevent any movement of water or salt. This is not true. It would shrink. Yes, as we saw that. It shrinked, okay? Because the water would flow toward the more concentrated solution. Okay? It says it would shrink. Let's read the others, please. It would swell. This is not true. As we said, it was shrinking. It would shrink. Yes, it may be. Because water would flow into the cell. It shrink, but water doesn't move into the cell. As we see that, water goes out, guys. That's why it's shrinking. So it's not because of water moving into the cell. It would swell. Swelling is it's not true. It's incorrect with the answer. So the possible answer for the question is B. Okay, let me clean the screen, guys, for you. So the possible answer is B. It would shrink because water would flow toward a more concentrated solution. More concentrated solution means to the hypertonic solution. It means to the high solute and low water. Okay, so this is the end of the part 1 of the cell. We will continue in the second part in which we have another 10 questions related to the cell.